I am glad to, I'm glad to see such a good turnout for such an important topic. It is my pleasure to introduce Richard Swanson, MD, a HAS member and a longtime member of the Science, Medicine, and Technology Committee. He is an internist with a specialty in rheumatology and immune-related diseases, many of which are related to our DNA and RNA. He attended Creighton University School of Medicine and trained at Alameda Highland Hospital and the Naval Regional Medical Center at Portsmouth, Virginia. While in private practice, he was on the faculty as a professor at Indiana University, the University of Illinois, and finally, West Michigan Stryker School of Medicine in Kalamazoo. And now, as they say on TV, here's Richard.
Can you hear me now?
Now we can. Yep. Great.
Okay. Well, good morning. Thank you, Bruce. As Bruce said, I'm Richard Swanson. I'm a retired rheumatologist. And this topic, CRISPR, came up at the SMT committee. And we were supposed to have a professor from Hope, who is a microbiologist or biochemist, but she had indicated her teaching responsibilities and her research responsibilities this summer were too great to allow her to do this talk. So I took it on, and I can tell you, I had to do a lot of basic research to understand this. And I'm still not sure I totally understand it, but CRISPR is not a drawer in your refrigerator. I think it's kind of a cute name, but it is basically a, now let's see here.
There you go.
There we go. It's a gene editing tool. Scientists have been using gene targeting to introduce changes into specific places in the genome by removing or adding whole genes or single bases. Gene editing techniques have been developed. The idea is that we're going to modify the gene to either improve our lives or to remove a disease process. It's been going on for quite a while using a couple of these other eponyms, ZFN, TALENs, and now CRISPR-Cas. We'll explain CRISPR-Cas. And it's interesting because every research scientist who was working on this gave it a different name. They called it Cascade, they called it CRISPR, they called it all kinds of other things. But the CRISPR-Cas system was found in bacteria and developed in the laboratory. It's currently the fastest, cheapest, and most reliable system for gene editing. Now, in this talk, I'm going to try to elucidate this system for you so you can see just exactly what it is. This is a picture of Jennifer Doudna from the UC Berkeley on the left and Emile Carpentier from the University of Paris on the right. They received the 2020 Nobel Prize in Chemistry for their discovery and development of CRISPR, and it's an application to the human as well as any other genome. CRISPR stands for Clustered Regular Interspace Short Palindromic Repeats. Now, what are they? We're going to come back to that in a later time and I'll explain to you. But basically, I'm going to start with some basic material so that we're all on the same page. It's very important that we understand because this is all new material and it is very confusing because of the names and because of the techniques. What is a cell? A cell is a fundamental unit of life. I just rubbed my left forearm and I'm sure I brushed off millions of dead skin cells. So the body is made up of tremendous number of cells. It's the fundamental unit of life. And the stem cell is our basic progenitor. And as you can see, it can turn into all the other cells in our body. So this is the basic mother and father of every cell in our human body. It basically, this is what we're all made of cells. Now, what is a genome? This term was coined in the 1920s, so it's not an old one. It relates to the entire set of genetic information found inside a cell. So everything about you or I, an animal, a bacteria or such like that, is contained in the genome. It's identical from cell to cell in any given organism, whether it's bacteria, a toad, mice, or men. Not all the material is useful in that cell, but that cell contains all of the genetic material that we need for that person or being. What makes up a cell? Well, a cell is made up of the cytoplasm, which is out here in the light gray, and the nucleus, which is down here. The nucleus is like the captain of the ship. It controls everything else. We're not going to spend any time talking about the cytoplasm other than proteins are made out in the cytoplasm. The nucleus controls the activity of the cell. This is a picture of the nucleus. There's lots of material in it, and we need to know this is where we're going to focus all of our attention today, on the nucleus. What's in the nucleus? Well, there's another organelle called the nucleolus, which we won't talk about today. But in the nucleus is most of the genetic material of the cell in long linear strands called of DNA, desoxynucleic acid. These are in chromosomes. If you took a cell, let's take a skin cell, open it up and take the chromosomes out of that nucleus and put them end to end, we'd come to about 2 feet. So you can see this cell is a microscopic thing, and all of this material is jammed into that nucleus. It's amazing how they can pile all this in. Now again, what is a chromosome? These are long strands of DNA and proteins organized into genes. Genes, not from any department store, are segments of DNA in a specific order that contains the code for a specific protein that functions in one or more of the cells of the body. In the human, there are 23 pairs of chromosome, one from your mother, of each pair, one from your mother, one from your father, and then there's the one set of chromosomes called the sex chromosomes, or the germline. This is a picture of human chromosomes. You can see that down here at the very bottom, there's an XY, which is a man, XX is a woman. But all of these other chromosomes, they're numbered, and some of them have very long, some of them are very much shorter. But this is what is in the cell of a human person. DNA. DNA is the chemical that's most important in these. It's made up of a sugar, a phosphorus, and 4 nucleotide bases. Nucleotide bases, adenine, guanine, thymine, and cytosine. It's in a double-stranded helix. This was the work of Watson and Crick back in the 50s to elucidate the structure of the DNA. And it was quite interesting to read, because they were battling Linus Pauling, who was sure that he was going to figure this out and get another Nobel Prize. But Linus got distracted by his political involvement, and Watson and Crick, along with Rosalind Franklin, actually found the double-stranded helix, which I'll show you a picture of here in a second, if I can get this to work. There we go. This is just a graph of what it's like. The base pairs are on the inside of this double-stranded helix, and they match up. So adenine always goes to thymine. Guanine also always goes to cytosine. So these pairs, or if you have either strand, you know what the other strand is by looking at what the base nucleotide. These outside, the blue part here you see, is made up of phosphates and a sugar. This in between is held together by hydrogen bonds, which is a very not very strong bond, which can be broken apart. So that when your cells divide, this DNA, the chromosome, divides in half. One half goes to one cell, one half goes to the other cell, and then they repopulate. RNA, you've heard all these terms, ribonucleic acid. It is similar to DNA, but it has many other purposes. Remember, the DNA is in the nucleus and it stores the information. How do we get it to where it needs to go? RNA does this. It takes the genetic code from the DNA to an organelle outside in the cytoplasm called the ribosome, where proteins are made. We have proteins are the basis of all of the body. And if you look at this, you can see that DNA is a double-stranded helix. RNA is a single-stranded helix. And as we'll show, there's one difference. As I said, RNA is a single strand. RNA is made up of 4 base pairs, but uracil replaces thymine. So on the RNA, you'll not have thymine nucleotide, you'll have uracil. The RNA base sequence is exactly the same as the DNA sequence. RNA can be found in the nucleus, but it also can get out of the nucleus into the cytoplasm, where it codes for the various proteins used in the body. That's not body. RNA, there are several kinds of RNA. Interesting, messenger RNA, it carries the information from the genes on the DNA to the ribosome, which is in the cytoplasm, outside the nucleus. It's the cell's protein-making machine. mRNA, interestingly enough, was what is being used to make the more powerful COVID vaccinations. So there really is no human or material in this vaccination that comes from a human itself. It comes from the mRNA of the virus. The second form of RNA is called the transfer, or tRNA. It takes the data from the mRNA to the ribosome and finds the amino acids that are needed and places them in the right sequence to make a protein. Ribosomal RNA, along with some special proteins, this RNA makes up the ribosome, which reads the sequence brought to it and places the amino acids in the right sequence to make a specific protein. Proteins make up the structure of the organism or of our body. Keep in mind that there are many, many, many DNA sequences, and there are many, many RNA sequences, but there are only 23 amino acids that can be made into a protein. Let's take a look at that. What is a gene? Gene is an area on that DNA molecule. Remember, if we go back, you saw those little black lines across the chromosomes. Those are the site of a gene. It's an area that's made up of a specific order of base nucleotides, which is the code for a specific protein, which may be the structure of the body or for certain diseases. I put down here a simplified gene just to show you that it could be base pairs of the A, CT, CCCG, so on. But keep in mind, many genes are thousands of base pairs long. So these are not 1 or 2 bases. These are long, long portions of DNA. In the human body, there are 3 billion base pairs, but there are only 20,000 genes. So it tells you that a lot of the space on those chromosomes is unused. It's just nonsense, DNA. It doesn't show us anything at all. Now, let's move on. Each gene is located on one or more of the chromosomes, like a pearl on a pearl necklace. If you look at here, this is the chromosome, and this is the DNA strand, and each one of these pearls is a set of DNA bases that code is a gene. So genes are specific areas on the chromosome that code for a protein in our body. Now, a major breakthrough in our understanding of human biology occurred with the Human Genome Project. This was an international project to identify and map the DNA sequence of every gene on all of the chromosomes. You can think about this. If we had some of these genes that had over 1,000 bases, how much work to identify in the exact order. This was already started by the US government, but a private company took over and said, hey, we can do it cheaper and faster. And they did. And this whole catalog, so to speak, was completed in 2003. Without this, we could never have proceeded the way we are now. This allowed us, let's take a look at this diagram down below. You can see these different markers, areas are different genes that have been identified. And so we know where they are on each chromosome. If you want to find gene X, you look on chromosome 15 and you know it's there and you know where it is. This allowed us to know where every gene was located and the actual DNA sequence of that gene. Without that, we couldn't have gone anything like we've done now. Now we knew where the gene was located, and now we could begin to edit or modify the genetic code. We could add a gene that allowed corn to grow in drier climates, or delete or modify a gene for such things as sickle cell disease, for cystic fibrosis, et cetera, et cetera. So this was a technique to go in and modify the human gene, since we know where all of the mutation genes are. So if you took my DNA, I do not have any genes coding for cystic fibrosis or sickle cell disease. But we know where those genes are. So if we can get to those genes, we may be able to modify those genes and prevent that disease from continuing to spread or to develop. Here's where we turn to bacteria. In 1986, a scientist began to sequence a gene in the E. coli bacteria. Remember, he was sequencing one gene. He found that 1,038 base pairs of DNA made up this gene. He found that in this gene, there were 5 sequences of 29 base pairs that sprinkled between the normal-looking sequences of DNA, and he called these normal-looking sequences spacers. But each of the 29 base pairs were identical and were the same backwards and forwards, a palindrome, such as the word Bob. Other researchers found these repeat segments in 20 other species of bacteria, which gave us the notion that this may be useful, and it will talk about it because it was the bacteria's defense mechanism against viruses. Again, clustered, regular, interspaced, palindromic repeats. So these segments could be looked at, these special segments were always the same. On either side of these repeats, however, were genes associated with Crispr. So we found that these genes went through multiple names, and we now started calling them Cas, CRISPR associated. These other genes, which are on either side of these repeat areas, coded for an enzyme, which we called Cas. And we'll find out these enzymes are the scissors in the cell that cut the DNA apart. When we first started working with Cas1, and then we started realizing that wasn't working in this area, so we started looking for more. Now there are over 13 different Cas enzymes known. So and what happened, for example, in the laboratory, they would take the Cas gene, or whatever the other Cas they wanted, plug it into E. coli, which is our favorite laboratory friend, and the E. coli would just start making, making Cas genes. It would just, thousands and thousands. They would take the bacteria, break it down, and extract the Cas enzyme, which we would use then in the laboratory in experiments. Since we knew the base sequence of every gene, and we knew where every disease gene was, we could go back to that area and work with those genes by making new genes, or RNA, particularly RNA, in the laboratory. So this was all being done in test tubes. This was just unbelievably difficult to do. You couldn't see it because it was so microscopic. Now, what did bacteria find? Well, bacteria have been invaded by viruses since time immemorial. These are bacteriophages, are viruses that attack bacteria and use them to make their own viral DNA. This is the largest group of viruses known to man. And I put this down here. These are pictures on the left and on the right, but the one virus we know of looks like a lunar lander. It lands on the surface of the cell, sends down a probe through the middle. Here's the probe going into the cell and releases its DNA. Remember, viruses have no way of replicating their own DNA. They've got to latch on to some other cell, a vector, we call them, and inject their DNA into that cell and order that cell then start making their viral DNA. It makes the bacteria make the viral DNA and viral particles. The bacteria then begins to swell with all this new material is being made. It ruptures, releasing many viruses, and that's how viruses spread. So in the human, for example, let's just take a human. The virus invades the respiratory cell, lining cells, manufactures more viruses. Those viruses are released, and it spreads to other cells, and now we have a disease state. The bacteria dies from this rupture. So what this was meant to do is keep the bacteria from dying. So they developed this immune system using these phages. The Crispr molecule and the associated Cas9 search for the viral DNA and cut it out of the bacteria DNA. The Cas9 enzymes are like a pair of very sharp scissors. The problem had been in the past that many of these other techniques weren't as precise. They may cut a little to the left, a little to the right. They may only cut part of one, but these Cas enzymes would completely cut both strands of the DNA. The bacteria, though, was smart. It kept small portions of the viral DNA in its genome so they could recognize that same virus if it tried to invade again. These snippets of viral DNA are called the spacers that we mentioned before. They're between the CRISPR sequences. Now, because of that Human Genome Project, we know the sequence of every gene. We can manufacture an RNA molecule, which is complementary to that DNA segment that we want to cut out. So if we say, okay, we want to cut out a segment of bases on the 14th chromosome. We can make it, we know that gene, we know that DNA sequence, we've got that, look it up in our catalog, and then we say, okay, we can make RNA. We know this has to be complementary. So for every A on the DNA, we're going to have a T, and we go on our uracil actually here. So we go down and we make these RNAs, and we attach the Cas9 enzymes that we have in a beaker to this RNA, and we add another little RNA called a tracer. We introduce this into the nucleus of the cell that we want to modify. The CRISPR-9 system goes to work, cuts out the sequence of the DNA that we want. This sequence may be the sequence that codes for disease such as sickle cell disease. For example, in the beta globulin gene, beta globulin is part of the hemoglobin complex. But beta globulin gene, we find that an A has replaced a T. So we take our CRISPR and go in and just set up so it just cuts out this A and replaces it with a T. Well, this will restore the production of normal beta globulin and then normal hemoglobin. So this is a way of modifying so that the person does not have sickle cell disease. Now, I want to keep in mind, this technique works only on single gene defects. For example, diabetes has about 20 different genes that have to work together to get diabetes. It is not amenable to this technique, or if it was, it would be extremely cumbersome. We would have to take each gene at a time and remove each one of them by making these RNAs and this stuff, and it would be extremely, extremely difficult. Schizophrenia is another one we've looked at. Interestingly enough, the James Watson and Crick, had a son with schizophrenia, and he was absolutely hoping to find a DNA sequence that would cure the schizophrenia. Well, it didn't happen. Okay, this process of cutting out segments has only been done in single cells and has been attempted in cells of living creatures. For example, in the beagle dog, this is a famous example, they've changed the gene for muscle mass so that we get an Arnold Schwarzenegger looking beagle. It's just big and muscular. The work had originally been done only in bacteria. Now they were trying it in humans, and it has been done in many, many other organisms, including many plants. We'll talk about that in a little bit later, because this is different than what we've heard about the GMO. Now, what this techniques that these two developed would allow us to cure certain human diseases. But remember, these diseases have one gene mutation only. Now, let's look at a mutated gene of 29 bases or less that was associated with disease and modify it. We cut it out. We either put a new gene in or we just leave it out. This could be done in minutes. Actually, they said it was amazing to watch these CRISPR genes go chomping through the DNA, and one of them was doing it like 3,000 cuts in a second. The zinc finger nucleotide process, which is the one they had before, was not always accurate, and sometimes we got the wrong things, and it took days to prepare, and the CRISPR would cost only a few dollars to do, whereas the other one was costing thousands. This system for cutting out the abnormal DNA was precise. It was easy to prepare and was much less expensive. They could pick out a specific DNA sequence from the 3.2 billion gene pairs or base pairs in the human chromosome. 3.2 billion. And this technique would allow them to identify any one. They could inject this process into human embryos, and it was spread to all cells and be transferred to all human genetic offsprings. This was in the gene line. This became very controversial. We'll talk about that. GMO. You've all heard of GMO products, the wheat, the potatoes, the tomatoes, the strawberries. People are say this is Frankenfood, and you're playing with nature, and they're claiming that this is not safe. They claim that it'll lead to cancer, et cetera, et cetera. None of this have been found, but they use splicing. They don't cut out, they put new genes in. And these genes may be totally foreign. CRISPR, in contradistinction, does tiny alterations in existing genes by tweaking the level of proteins that were there already. The CRISPR is degraded within hours and leaves no trace. So after it's done its job, there is no evidence in it, in the cell, in the nucleus. This is called scarless gene editing. So it is a technique that was developed and in 2015, basically, and in 2020, they got the Nobel Prize winner. Keep in mind that only two people get the Nobel Prize, and that this was the first time that two women got the Nobel Prize. There's always been, two males and a woman, or two women and one male, but this was the first time that two women. Now, moral and ethical questions were raised from the very beginning, and many of these scientists had begun working in somatic cells, changes that would affect only that one organism, you, me, or Joe down the street. The 23rd set of chromosomes were called the germline. This controlled the sex, and these changes that could be done in one egg or a sperm would be passed down to all future generations, opening the discussion for, quote, designer babies. I want a male, blonde hair, blue eyes, with an IQ of 140. Let's go to work. This was dangerously close to the eugenics movement of the 1920s, which we heard about, or the Nazi superior race ideas. They were doing it by using natural selection. They had no idea about gene cutting, but they were trying to breed people of certain nature and get more of that, or even better. This was considered very unethical, very dangerous, and it was considered altering what 2 billion years of natural evolution had accomplished. Should we try to control the future without knowing the consequences? That was a very serious question that they asked. And by 2015, most scientists agreed that we were not going to be working on the human genome, human gene line, germ line. It had not been fully elucidated and was not ethical. However, there was always that rogue scientist, and this was in China, Huang, injected CRISPR into human embryos to prevent beta thalassemia. 4 embryos were viable after they did this. 2 were implanted, and there are twin girls in China who carries this. This work was published by Huang, kind of just blowing everybody else's mind apart, and there was a huge concern in the scientific community that this was unethical, and the response was swift and unanimous. He had stepped over the red line of ethical research. Huang expected to be a hero in Japan and China. It turned out that even the Chinese government chastised him and told him that this was not the thing to do. And since that time, there have been a number of international conferences which have dealt with this. And we've decided that we must differentiate between gene therapy and gene enhancement. The Designer babies were gene enhancement. Social justice issues came up. What could happen since this was not, I mean, it was theoretically only a few dollars, but in practicality, it would be costing hundreds of thousands of dollars. What they would have is then there would be inequality. We'd have some people who could afford this and would be saying, hey, please take our babies and make them into the superior race. We didn't know the consequences of this enhancement. What would happen 2 generations down? Would these people all die of some form of cancer because of this changes? We didn't know. The religious issue came up, and this is straight from Genesis. God made man in his own image and likeness. If we changed man, into whose image and likeness would he be? That of the scientists? Would have that may not be image and likeness of anybody? This raised many, many theological issues, and we realized that gene enhancement would create even greater disparities between races and classes. Can you see that in India, if all of the Brahmin classes were enhanced, and there would be even greater disparity between them and the other castes in India. So far, right now, many countries have banned research and clinical trials in the human gene germ line. The U.S. has not done so. We were somewhat hesitant, and when you read the U.S. Report on this, it's very tippy-toe around this. But the U.S. has passed, the government has passed a ruling banning the funding of any research that would destroy or create human embryos. So we could do research on bacteria, pigs and mice, cows, but we're not going to do it on humans. So far, they have done this, and many of the successes have been in the animal world. We know that cows are in quarantine, in captivity, in feedlots, for example, take up a lot more space if they have big horns. So we have bred cattle that don't form horns, giving them less problems in the feedlots. We have used this on mice. Of course, mice we need for research, so we've got to keep breeding mice. So we've been able to change some of these mice and to make them different for this or that different kind of research. We've done this in food. We've done this in all kinds of other creatures. And very interestingly, this technique was really first brought to Dudna's attention by the Dannon yogurt people. They were having trouble with many of their bacteria that they used to make yogurt and other dairy products weren't working. And it turned out they'd been invaded by viruses and had been deactivated. But they found that they could, certain of them could be treated, and they with CRISPR, and they would survive. So actually, this technique has been around and used in different animals and creatures, but it has only been done in somatic cells in humans. There have been several experiments using this, trying to put these in to change various human ailments. Some of these have been successful. Some of them have an absolute disaster, and people have died. They injected a couple of people with CRISPR, and they developed massive leukemia and died. So you see, we do not have all of the answers, or can we control the consequences? I want to end this with the power to control our species' genetic future is awesome and terrifying. Deciding how to handle it may be the biggest challenge we ever face. How do we do this? Scientists, as you know, are being viewed with increasing distrust by the public. We've seen this with the coronavirus. The public is skeptical about our contribution to society. And when people refuse to accept climate change reject vaccination programs for children, it signals not only their own ignorance, but also a complete breakdown of the communication between the public and the scientists. And what this talk was meant to do was for the general public, which is HASP is made up of a cross-section of all walks of life, is meant to facilitate this communication and lead to better societal acceptance of what science can do. With that, I'm going to end, and I'll open this up for questions. So I'm open for any questions that you have. I hope that I've tried to explain this enough. Obviously, what I've done has been very superficial, but it is an overview. Now, if you want to get into it, there is a whole book. Jennifer Doudna has written a book called Kraken Creation, which is her own book. And then Walter Isaacson, who is a well-known author, has written another one called The Code Breakers. And it's about 400 pages. So if you want to get to know everybody who's involved in this and what they've done and where it came through, you can read these books. Besides that, there is now a journal of CRISPR. You can go to it online or subscribe to it if you're so inclined. But I can tell you, trying to read some of those articles is absolutely impossible for those of us who are not in that basic science category. Thank you very much. I'm open for questions.
Okay, let's see, Richard, I have one in the chat here that says, you said that only single gene diseases have been attacked by CRISPR. Are there any diseases that involve only a couple genes? Could they be attacked?
I'm not aware, but many cystic fibrosis, sickle cell disease, a couple of other combined immune deficiency diseases are one gene. Multiple genes have not been attempted because they don't know if this technique could be done over and over again on the same cell. So no, it hasn't been done on diseases that have 2 or 3 genes involved.
Okay, thanks. And then I just have a request from Diana to let people know that a little plug for the HASP nonfiction books class in September. They will be discussing The Code Breaker by Walter Isaacson about Jennifer Doudna.
That is a real eye-opener, and good luck on those who have to read it, because it is complicated. And remember, she's a research scientist, so she's not a clinician. And so it's difficult at times to keep track of what's going on. And to me, I've always been amazed that they're doing this stuff in test tubes. So they open up the refrigerator, take out a vial of Cas9 and put a few drops into this and do that. So it's all very science fiction-y like. But good luck. It's a good read. It's very difficult, but Isaacson writes in a better style than Jennifer herself does. So it's better than her own book.
Thanks. That's all I have in the chat. So if anyone else wants to unmute themselves and ask a question or make a comment, please feel free to do so now.
I have a question, Richard.
Yes, sir.
I understand that obviously this happens in the nucleus. And as you were describing it, you were saying that the CRISPR molecules were placed in the nucleus. That works great doing one cell at a time. How do they treat cells scattered all over the body?
Well, what they do is they'll do this, say, in an embryo. And then these cells, remember, we talked about stem cells. So that goes from a stem cell and it continues to grow and basically takes care of every cell in the body that has this defect. This has been the problem, Bruce. When we've done some of this, it didn't take. So some of these people only had maybe 1/2 of their cell was modified, of their cells, and the other half wasn't, which left them kind of in a quandary. Do they get 1/2 of them get cystic fibrosis and half don't? We don't know. That's a problem, making sure that it's going to get to all cells. Now, in bacteria, which we did this, it was easy because you knew they grow and you could see them in a test tube.
Sure. And there's only one cell there to start with. Sure.
That's correct.
So this treatment is really for embryos at this point. It's not immediately applicable to adults because of
It
this.
has been done in adults. Some adults have been done. We have done this in taking out the bone marrow and getting the stem cell and treating it and putting it back in. But it's not been as successful as it looks in theory. I can give you a list of diseases that are here. Let me get it. That are amenable to this techniques. I gotta find it here. Okay, here are some of these. Duchenne's muscular dystrophy, certain cancers, achondroplasia, which is a dwarfism, chronic granulomatous disease. There are some views about Alzheimer's. They think there's some Alzheimer's, not all. They're a single gene. ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease. Tay-Sachs disease. These are all possible CRISPR diseases. Sickle cell anemia is another one. And this has been looked at with great and has been done in several people and with good success. But because it's working with humans, we're very, very careful. And the human experiments are really experiments.
Richard, are
Yes.
there possibilities for things like Parkinson's that are more chemical depletions?
Well, remember, chemical depletion is based upon a protein. And sure, in the gene, in the chromosomes, there has to be a gene that codes and makes the wrong proteins, or doesn't allow proteins to be made. So yet we have to look. Parkinson is, as I know, has more than one gene site involved. So it's not totally open. We may be able to partially correct it by going after one of the genes, but we right now it isn't high on their list.
Thank you.
Richard, this is Bill Wells.
Yes, Bill.
Your presentation gave me more information on this subject than I had been able to gather from any other source. I don't know how to put this in a different way except to say that you're sort of the Walter Cronkite of our Medicine, Technology, and Science Committee.
I'll have to get some big black glasses frames. I used to smoke a pipe, so I'll have to get that out if I can find one.
Your method of presentation is somehow informative and believable and all in one bunch.
Thank you. This is what I tried to do. I mean, this is how I learned. And I can tell you that I was rereading some of this material until 2 o'clock yesterday afternoon.
Fantastic. Well, you must have done hours of research.
I'm tired of these books. I'm going to put them down and go back to some fiction.
Well, thank you. Thank you for today.
Yeah,
Yes,
we really
indeed.
appreciate your efforts.
I don't know, am I hooked up here?
Yeah, go ahead, Marshall.
About a month ago, there was a report by the corporation Intelia. This is the company that is formed essentially by Jennifer Doudna and her colleagues, where they report success in treating humans with a liver disease, an amyloidosis, in which the liver overproduces an amyloid. And they reported success in, they only treated about, I think it was 6 patients, and there was relief from the disease in all of them.
That's correct. There's another disease called alkaptonuria, which also is a single gene, but the liver disease with amyloid is exciting because amyloid is a proteinaceous material, and it can be, amyloidosis is a terrible disease, often occurs with other chronic diseases such as rheumatoid arthritis. So if we can prevent that, we're going to make the life of these people a lot better. So yes, they're doing it with experiments, but yes, you talked about Jennifer. She founded several companies, for-profit companies, to do this. And yeah, you can go online here now and order. I did this too. I went online and found I could order Cas9 enzymes. Do I want a small vial or a big vial? So, I mean, the idea of this is to make it so that it could be done in the average laboratory, not in one of these super laboratories like at UC Berkeley.
But the exciting thing about this particular experiment was that they injected, as I understand it, the reagents into the bloodstream. They found their way to the cells in the liver in an analogous way to the way the mRNA vaccines are given, that is in liposomes.
Yes, yes.
Inject and find themselves.
You understand. I did not bring that up, but that's again more detail. But what you're seeing is exactly what we're doing. We're doing very slow and methodically, and it's being done by for-profit companies. And that's why it's not cheap right now. They're making a profit, and they're making Jennifer and a few of these other people quite rich.
They're also making people who buy the stock pretty rich.
Yes, I think that too. I don't know much about their stocks. I'm not sure where they're traded, but somebody else can tell me about that stuff.
There are a couple other ones that came in the chat. Maybe you answered these, Richard. One is, has CRISPR been attempted for Alzheimer's? And the second is, was CRISPR used to create the COVID-19 vaccine?
No, CRISPR has not been used to create the COVID. We've actually used CRISPR to try to detect COVID and do a better job of detecting it in the body, better than the antibody test that we have now. It has not been used to make the vaccines, but it has been used to detect the COVID virus.
Okay.
Now, Alzheimer's, you asked about Alzheimer's. I know there's research in this, but I'm not aware of the actual status of it now.
Okay.
Don't you think the most likely diseases to be actually cured will be beta thalassemia and sickle cell anemia?
You're absolutely right. You know, we know that sickle cell anemia is a huge problem in Africa. The only problem is that these people are protected against malaria. Now, we've used this technique to treat mosquitoes so they don't carry the malaria germs, so that we can probably go at it from two different directions. But yes, you're absolutely right. Beta thalassemia and sickle cell disease are very high, and they have been done in some experiments, and again, some very successfully. Well, I'm going to end. It's time. It looks like it's time, 10:30, Kim. But I want to thank all of you for coming. I can tell you that I have learned so much doing this presentation. I'm not sure how, as a clinician, this would be a benefit to me yet. But the idea of this talk was to spread the knowledge so that when you hear it on the news, you hear the news people talking about this, you know what's going on. Thank you so very much.
Thank you, Richard.
Thanks, Richard.
You're
Thank you
welcome.
very much, Richard. Great job.
Great overview. Thanks, everybody, for being here. Enjoy the rest of your day.